Okay, everyone, we have another great session coming up for this meeting that is going certainly better than I believe most of us expected. Expectations were high, but it's going very, very good. So our next speaker, who I tried to imitate a minute ago by yelling everyone to come in, is Mike Friedlander. Obviously doesn't look anything like me. His accomplishments obviously are nothing like mine. Mike is the host of this meeting. He's also the co-organizer of this meeting. Michael, as all of you know, is the director of the Virginia Tech Fralin Research Institute, and recently he was appointed as the vice president of health sciences of Virginia Tech, sciences and technology of Virginia Tech. So Mike wears a lot of hats as administrator, has made a lot of contributions also as an educator to the scientific enterprise, but also to promote science in Congress, participating in panels and other entities. But Mike has been doing what he loves most over 30 years, and that is asking what is the organization, but also the function of neural circuits in the mammalian visual system. How do they organize? How do they function? He has made tremendous contributions that are exemplified by many publications in top-tier journals. So what I'd like to do is introduce Mike Friedlander, and he's going to tell us today about the role of pattern stimulation in inducing synaptic plasticity. Mike?
Thanks, Greg. Appreciate it. Great to be here. Okay, well, now I get to take off some of those other hats and just be a regular scientist, which is the most fun. And I'd like to share with everybody a little of the work that's been going on in our lab over the last couple of years. So first I'll just share with you, here's where you are. This is part of the Virginia Tech Biomedical complex. Just in case you don't know what that is, it's called a hokey bird. It is the mascot of Virginia Tech. It's some sort of a turkey on steroids, basically. And the other thing that's really fascinating about the hokey bird is I think Virginia Tech is the only university in the United States that at their football games eats their mascot. So they serve cooked turkey, fried turkey legs at the football game, so it's quite a spectacle. Anyway, welcome to the beautiful area around here. We're only a couple weeks away from this sort of color. I wish you all could stay and see some of that, but I know our Nordic friends will be seeing plenty of that at home and the beautiful colors back there as well. So I'm going to talk a little bit about our work with respect to plasticity and brain injury, but I want to first, not sure what's going on here, Jerry. Oh, what's going on is I got the wrong one. But I first want to acknowledge the people who do the work, and you all know this drill. These are the people that really do the work. And so primarily the work I'll talk about today will be, that I'll emphasize, will be done by Quentin Fisher and Haja Kalakulov, both of whom are in the audience. But I'm going to touch on some of the other work that's been done by some of the others here who have been in the lab before. Ignacio Says and Ishgander is here, a former student Olivia, and Susanna, who's here as well. And the work is, of course, supported by the funding agencies, NIH, and in this case, the Department of Defense. So I think in honor of our Nordic guests, I wanted to make sure to get this slide up there. Jan Storm Mathisen, I want to make sure you see this slide. So I've been educated to learn of the importance of a number of Nordic investigators in neuroscience, and of course Fridtjof Nansen from the neuron to the North Polar Sea, who's a Nobel Prize winner for the Peace Prize, actually, recognized, but maybe not recognized enough for appreciating the importance of the neuron and the structure of the nervous system. A little quote here about why he wasn't perhaps recognized as much as he should have been. But I've been educated and very aware of the amazing contributions that Nansen has done, contributing to our understanding of the nervous system. And of course, we just heard yesterday about the Brain Prize winners for this year, and that relates a little bit to the subject of my talk, because I, like many, have kind of picked up on a lot of work that these very, very famous and productive people have done with respect to understanding plasticity in the brain. Now this is a classic, I love to show old slides, maybe it's because I'm that old. A classic old diagram from neuromuscular junction physiology from Boyd and Martin. And of course it shows a classic recording of an EPSP and a muscle cell stimulating a motor neuron. And what you see here is the huge safety factor. That is, when a motor neuron, alpha motor neuron, decides to fire, the muscle's going to go. You're going to get an end-plate potential, an action potential, and the muscle's going to contract. Well, that's a good thing, right? You don't want your neuromuscular junctions making decisions and being particularly plastic, like maybe I won't move now, even though the brain told me to move. So we kind of get that, and as I said, that's a very good thing. However, in the central nervous system, depending on where you look, synapses are a lot more fickle. This is a picture from some earlier work I did with my colleagues Yves Fragnac and Steve Redmond down in Australia, Yves from Paris. And we were doing some multiple patch clamp recording in slices and visual cortex and recording from pairs of neurons. And in this work, what we saw very dramatically is if you take the classic textbook view of an evoked synaptic response from one cell, it's very smooth when you average thousands of trials, of course. But when you look on a trial-by-trial basis, you see quite a lot of variability in the small responses, and of course, sometimes you see total failures. And this can be dynamically regulated, plasticity, functional plasticity to synapse. Best characterized probably by the many, many studies on LTP, long-term potentiation, and LTT, long-term depression, and various stimulation protocols that can induce these long-term changes in synaptic responses. And so in our lab, we've been trying to understand a little bit more about this in the neocortex, particularly in the visual cortex. Of course, the hippocampal formation is very well studied in this respect because it's an area we know clinically and from basic science is very important for the formation of memories and learning. And it makes a lot of sense to study synaptic plasticity there. Does it make as much sense to study synaptic plasticity in other parts of the brain? I and others would argue yes. Virtually every part of the brain is dynamic in some respects. And we happen to be interested in the visual cortex, and we study these processes in that circuitry. And so several years ago, we published a paper that Iskander Ismailov and Haja were lead authors on, where we were trying to do some just simple experiments where we paired pre- and post-synaptic activity to look at plasticity in the visual cortex. And we did the experiments, and many times they came out the way we expected it, and we paired activity. This is a classic example of long-term potentiation. But sometimes we do the same experiment on a cell right next to another cell that for all the world we thought was the same cell type. It's in the same layer, it looks the same, it behaves the same electrophysiologically, and we do the same plasticity induction protocol and nothing happens, no effect. Or sometimes just the opposite would happen. The cell would show depression versus potentiation even though we're using the same protocol. And so we went on to study using calcium imaging what was going on inside those cells. And to make a long story short, what was found was there was a nice correlation between the dynamics or the kinetics of the calcium signal in the cell and the plasticity outcome. So for example, if the cell showed potentiation or LTP, the calcium signal in the cell went up very rapidly. If it showed depression, LTD, the calcium signal rose more slowly. They actually reached similar levels, but the kinetics were different. And when there was no effect, when we did another type of control experiment, the calcium signal just sort of bumped around, but it didn't seem to have a very, very dramatic signature. So we know calcium inside the nervous system comes from a lot of different places. We know it's a double-edged sword. Calcium is very important for plasticity, learning, memory. It's also very important in excitotoxicity and cell death after a brain injury or a stroke. So the brain goes to great pains through buffering systems, pumping systems to regulate intracellular calcium. And this little cartoon just shows various sources of calcium for a neuron, various types of voltage-gated calcium channels, of which there are certain pharmacological blockers, certain types of AMPA-type glutamate receptors lacking a certain subunit will flux calcium, NMDA receptors, metabotropic glutamate receptors will cause calcium release from intracellular stores. And you can play around with all these pharmacologically and take one out of the picture and see what happens. So in a series of studies done primarily by Dr. Kalakula from my lab, he decided to look back at the same variability in plasticity that I already showed you. And this just shows a histogram showing the net outcomes for single cells. And the red show the cells that ended up potentiating, the blue, the ones that depressed, and the gray, the ones that didn't change. Same protocol I showed you before. And then underneath, they're showed a series of panels where pharmacologically, one calcium source at a time was blocked. NMDA receptors were blocked, L-type voltage-gated calcium channels were blocked, ryanodine receptors were blocked, metabotropic glutamate receptors, or intracellular IP3 receptor calcium released from intracellular stores. And you just kind of squint and look at the slide and you see what's going on. When you block certain calcium sources like NMDA receptors, voltage-gated calcium channels, or ryanodine receptors, you don't see any red. You shift everything to depression. When you block the metabotropic signaling pathway downstream to the IP3 receptor, you don't see hardly, except for one case here, hardly any blue. Everything is shifted to potentiation. So it's as if somehow a selective differential accessing of calcium signals can tip a cell one way or the other. Same experience, same activity. If I extrapolate for a moment, maybe the same thought, the same experience, and that neuron goes this way, and the other one goes that way, synapses up or synapses down. So when we start to talk about cell types in the brain, I would submit an important part of that program in terms of the brain initiative would be to think about cell types in terms of their plasticity behavior as well, because it's not one size fits all, and we can't take everything we learn from one cell type, say in the hippocampus, and apply it to every other cell in all parts of the brain. So there's precision and variability even at that level. So one of the questions we asked in our lab, and this was a dissertation by a graduate student, Olivia Fitch, whose picture I showed you before, was when these differential plasticity behaviors happen on a single cell, are they happening for all the synapses on that cell, or are the synapses different on an individual cell? So the experiments that Olivia did was to patch clamp onto a single visual cortical neuron, a layer 2-3 pyramidal neuron, which is, I should have mentioned, the neurons we've studied in the other studies as well. And then she would activate different synaptic inputs to different part of that cell, let them both participate in the same learning experience, if you will, the conditioning paradigm, and see what happens to different inputs onto the same cell. So this is a picture of experimental paradigm. It's a slice of visual cortex, so it's an in vitro acute preparation taken from a mouse. And she's patch clamp recording from a single neuron, a pyramidal neuron in layer 2-3. And then she has two micro-stimulating electrodes positioned in layer 4, stimulating afferents coming up here in different spots. The first thing she has to do is figure out that she's not stimulating the same inputs because of current spread. So it's a tedious, painstaking experiment where you move the little stimulating electrodes, you stimulate one and then the other and then stimulate them together, and you make sure you're activating different inputs. And then you stimulate them in an interleave fashion. Pathway A, wait 10 seconds. Pathway B, wait 10 seconds. Pathway A, 10 seconds. Back and forth. So you're getting baseline on the different inputs of the same cell. Now you've got your control, and now you want to expose them to a conditioning paradigm and ask if they respond dynamically in terms of their plasticity the same way or differently. This is just a methodological slide to show how we separate the inputs using a pretty old-fashioned electrophysiological paradigm called an occlusion test. And I won't go through it in details, but it works well. So she did the experiments, and here's what she found. In some cases, she found this, i.e., these are the baseline responses of both inputs onto the same cell, and this is what happened after the conditioning. They both went down. So they both showed long-term depression, or LTD. Now, if all the data came out this way, we would say, okay, a given cell, a neuron in the brain, is in a plasticity state. All its synapses respond the same way. And that would be one answer to the question we posed. But it didn't quite turn out that way. That was just one example. Another example here, and I'll show you the group data in a moment. Two different inputs of the same cell, both exposed to the same conditioning paradigm. One of them shows synaptic depression, or LTD. The other inputs onto the same cell that experience the same pairing of pre- and post-synaptic activity at the exact same time undergoes a completely opposite response, LTP. They go in opposite directions. So what did the data look like when you pull it all together? A couple examples are nice, but what did the data really show? We considered every possible outcome. That is that both inputs would show depression, both inputs would show potentiation, one would show potentiation, one depression, one no change, et cetera, et cetera. This is what we observed. These are the expected outcomes based on studying one input pathway at a time. We did the statistical analysis for all these pairs, and basically we found out that there was no correlation. It was random. And so, in other words, what we have here, we plotted the correlation analyses. We did a Monte Carlo simulation, ran this 10,000 times. There is no correlation coefficient between one set of inputs and another of any significance to tell you can predict what one set of synapses will be doing on a cell and another, i.e. the synapses are the independent units with respect to their plasticity behavior. So the repertoire computationally of a single neocortical neuron in terms of dynamics is even extended because each group of synapses can be acting in its own plasticity way, if you will. All right, well, those experiments are fun and they're nice, and we do them in the lab where you stimulate regularly, bump, 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 bump. But I spent 20 years of my life sitting in labs recording electrophysiological responses in vivo in brains of living animals and sitting next to a loudspeaker all night and listening to action potentials. And to this day, I have hair cell loss from that. I can tell you I don't hear so good in certain frequencies. But nonetheless, I'd go home at night, when I did go home, and I hear this boop, 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 irregular thing, and I said, you know, I'm sitting in the lab creating experiments going boop, 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 but brains usually don't go boop, 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 they go boop, 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 boop. And so it was noisy. So years ago, a few of us got together. My colleague Reed Montague was involved with some of these early experiments and several previous postdocs and students, and we started looking at sort of the noisiness of the signals in terms of plasticity. And then I carried those experiments forward with my lab here, and the fellows in my lab have done a lot more on this. And this just shows a recording in vivo of firing of a visual cortical neuron. You can see how irregular it is. And when we think about precision neuroscience, are we going to have to know something about the patterns of activity in your brain versus my brain to develop therapeutics and interventions that modulate physiological activity, or will it be one size fits all? These just show what are called dot rasters of responses of action potentials, trial by trial by trial, of a single neuron to the same stimulus repeatedly. You can see the variability in the response and how noisy it is. So everybody, I think, in this room is well aware of the growth of the industry of deep brain stimulation. It's being used or being talked about for use in so many areas now. Of course, Parkinson's disease is an area where it has dramatic effects and a very strong record already. A lot of other areas where there's some promising early results or ideas that this might work. So this whole idea of tapping into the brain and modulating the circuits through stimulation versus pharmacologically bathing the whole brain in some agonist, for example, that may end up binding lots of places you don't want, is really a cool one, right? Use the brain's own internal circuitry, release neurotransmitters from the circuits you're stimulating in a physiological way and modulate the brain, and maybe you can do lots of good with it. And so we have been interested in traumatic brain injury and some question about whether it would be applicable there. Of course, when you go to stimulate the brain, there's a lot of ways you can do it. We've heard about some of those this morning, some very elegant ways. There's transcranial magnetic stimulation, focused ultrasound now, optogenetic techniques, electrode implantation, deep brain stimulation, etc. And then once you stimulate, you got to worry about all this stuff, or you got to think about it at least. All these parameters: how long should the pulses be? How many? What's the frequency? What's the pattern, etc. And as I've looked at the clinical literature, my impression is people are starting to give some more thought to that, but there hasn't been a major systematic investigation of that parameter space. And so we thought we were going to take a step back and ask at a very basic science level what some of this means for the brain, particularly for the downstream plasticity effects that could happen. So in conjunction with this funding support from the Department of Defense, we began a study to look at this in injured brains with the idea that down the road, perhaps therapeutic intervention and deep brain stimulation or some other method could help strengthen and weaken damaged pathways where the brain wasn't killed, but where there was a change in the balance of activity. But we, of course, had to do the control experiments as well. So we did a systematic study in control animals. So this is the paradigm. These are done in rats. The animals anesthetized. As a cranial window usually gets an impact injury on the brain, although we've done some blast injury as well. 2 weeks later, the animal comes back, has behavioral testing, re-anesthetized. We make acute brain slices to do electrophysiology, patch clamp on the neurons, use various stimulation protocols, look at plasticity, compare different parameters in normal animals and in brain-injured animals. And we've done immunocytochemical analysis to look for various markers to tell us we indeed have a traumatic brain injury effects, such as the upregulation you see here of GFAP. And then the animals were checked behaviorally, and we saw what we would expect. We saw some changes in time for writing responses behaviorally in the mild traumatic brain injury animals. And as I said, we looked at a lot of different markers. I'm not going to have time to go into this in detail. Blood-brain barrier markers, markers for neurons and glia, but we saw everything consistent telling us we indeed had a brain injury. So here's the experiment. We take a train of action potentials, we're going to stimulate with these to the input, and then we morph the distribution from perfectly regular to slightly irregular to highly irregular but we keep the you want me to do that again? I've been working on that all night. We keep the frequency the same. So in one set of experiments, it's always 1 hertz, or it's always 10 hertz, or it's always 100 hertz. But we change the parameter of the variability by changing the coefficient of variation of the inner stimulus interval distribution. So we keep the number of pulses the same. We keep the duration the same. We keep the average frequency the same. We only vary the pattern. And this is just an example, if you plot for the different coefficients of variation from 0 to 1.0, what the interstimulus intervals look like. Perfectly regular, they're all the same. Highly irregular, approaching a Poisson type distribution. And so this just shows the protocol. We collect baseline recording from cells at a low frequency, 0.1 hertz. We then do the conditioning at some frequency and some pattern, and then we collect the post-conditioning recordings. And this is an example of what the data look like. Here's the baseline normalized postsynaptic response. This is during the conditioning, and this is after the conditioning. And then we measure the strength and fidelity of the response here and compare it to the beginning and see if it underwent synaptic potentiation, depression, no change. So that's a straight-up paradigm, a lot of parameters. I'll summarize them rather quickly. You're looking at the group data. These are the control animals across the top. These are the mild traumatic brain injury animals across the bottom. This is for 1 hertz, and this shows the different coefficients of variation, 0, 0.2, 1. Perfectly regular, somewhat irregular, highly irregular. All at 1 hertz, all control animals top, all brain injured animals bottom. As you can see, the place we found the biggest difference is right here. For the very variable responses, much like the normal brain firing, we get a dramatic different effect in terms of plasticity in the normal versus the injured brain. In the injured brain, we get the predicted depression, or LTD, as we got really at all the coefficients of variation, some difference in the magnitude, but basically same response. And in the injured brain here, we got a completely different response. First of all, highly variable, and secondly, a reversal, an actual potentiation versus depression. So a clear difference there between the injured and normal brain. These data are summarized here. I won't bore you with a lot of data, just call your attention to the right panel. It shows for the coefficient of variation the cumulative probability distribution of the synaptic plasticity outcomes, and they are different. We did it at 10 hertz, and again, due to time, I'm just going to cut to the chase here. Once again, here's where we saw the difference. With the irregular patterns at a coefficient of variation of 1, completely opposite response in the injured brain versus the normal brain. And this, once again, just summarizes the statistical data for that. And then we did something called 10 hertz discontinuous. Instead of running 10 hertz continuously, where you could fatigue the synapses, we had little burst epochs, but we still could vary the regularity within each burst. And once again, it didn't matter. Completely opposite response at coefficient of 1.0. The brain injured cells showed a very dramatic potentiation. And by the way, notice during the conditioning, they're depressing, just like the normal brains. But they bounce back after, and they're potentiated, unlike the normal brains. And again, this just summarizes here the cumulative probability distribution to show you that's statistically significant. And in each case, for the highly variable response, we got a statistically significant difference between normal brains and injured brains. Now, we wanted to take this to the level of single cells, so we also did an experiment. Zhangping Wu has done this experiment, and I realized I didn't put his picture up there. I apologize. Where he's done a similar experiment between individual neurons, not stimulating a bunch of inputs, but pairs of cells. So he patches onto multiple cells, looks for connections between them from cell A to B and B to C and C to A and so forth, records a single cell response. And again, due to the interest of time, I'm not going to show you each of those. I'll just cut to the chase. And what he finds is the same sort of thing. Using these irregular coefficient of variation 1 responses, these were done in normal animals, he finds very different responses. And the cool thing about recording from single cells is you can analyze what's going on mechanistically. And what he found the difference was that the failure rate for synaptic transmission had changed. So that you can do what's called a quantal analysis, so we can get down to a single cell level to understand the mechanisms of this. I'll leave it there because my time is out. So, in summary, what we found is that neocortical neuron synaptic plasticity is highly variable, even in response to identical activity. Calcium sources play differential roles in the induction of neocortical synaptic plasticity. Different synapses on the same neuron show different dynamic plasticity states and act as their own computational units. The temporal patterns of synaptic conditioning induce different outcomes in intact versus the injured brain, depending on the frequency, where the more irregular patterns trigger dramatically different plasticity outcomes in the intact versus the injured brain. And so we think that this could be due to a possible change in calcium levels. I didn't show you the data, but Quentin Fisher and Haja measured intracellular calcium and found that it's elevated in the injured brain neurons, and that may trigger them to the opposite type of plasticity. So we hope this information will be useful going forward as new technologies are developed to actually modulate the activity of the brain, and in therapies and things like brain injury, building on these data. Thank you.